Hello students, in this lecture we are going to study some assay which are useful for the detection of hERG channel. So several methods are proposed to investigate the Q2 prolongation, and it is important to screen the new chemical entity or drug molecule on hERG channel. So in the previous lecture or presentation or video, we have discussed about some introductory part about the hERG assay. So those who have not getting that video, please go through the YouTube link which is provided here. From that you will get the basic about the hERG gene. So now, as you know, the hERG is blocked by the drugs which produces the prolongation on QT interval in the heart and thereby increases the risk of torsades which produces or which worsening or which aggravates the patient cardiac condition, and that can lead to the sudden cardiac death. So the scientific studies have reported that the many drugs have failed in the late phase of clinical trials only because of cardiotoxic effect such as Q2 prolongation. Hence, it is important to identify hERG inhibitors in the early phase of drug discovery. This line is very important, that the identification of hERG inhibitors in the early phase of drug discovery, so that afterwards it will not produces any type of the QT prolongation, or the risk of QT prolongation will be reduces. So let's see the methods. The methods are divided into three parts. First is the in vitro methods. Under that, fluorescent membrane potential sensitivity dye method is there. Then radioactive ligand binding method, then fluorescence polarization ligand binding method, then rubidium flux method, and patch clamp method. Ex vivo, there is a Perkins fiber method and Langendorf perfused isolated heart method. In vivo, there is non-invasive telemetry and wireless telemetry system is used to find out the activity on hERG gene. So let's see one by one how these methods are useful. So first method is fluorescent membrane potential sensitive dye methods. So in this method, the very first thing is that the cells were transfected with the hERG gene. Then after this transfected hERG gene, these cells having actually the negative potential, these cells have negative potential which are transfected with hERG gene. Then this combination, they are pre-incubated with the test compound of your interest, and one dye. Particularly, this is the name of the dye, but this dye is used. Here you can observe in this figure, this is the cell and these are the potassium ion channels, and this is the dye which are added into this. Then after, once the dye is added, the dye flow in and out of the cell, and this movement is based on the membrane potential. Dye interact with the cytoplasmic components to increase the fluorescence. So inside the cell, the dye get interact with the cytoplasmic components, and thereby they will increases the fluorescence, whereas the dye outside or extracellular solution, it will not produces any type of the fluorescence. So here the drugs, these are the drugs, test drugs. If they having the capacity to bind the hERG gene, then they block this channel and membrane potential increases. Because of this binding, membrane potential increases, and thereby there is an increase in the fluorescence level. So it can be observed by using the fluorometer plate reader for 3 to 15 minutes. So this is the first test, that is fluorescent membrane potential sensitive dye method. Now the second method is radioactive ligand binding method. So here in this method, HEK 293 cells, they are transfected with the hERG gene. Then after they were incubated at 37 degrees centigrade for 30 to 60 minutes with the test compound and the radio label ligand. So this is the radiolabel ligand. So this process is quite similar to previous one. So there we use the dye, and here they use the radio labeled ligand. Then the radio-labeled ligand and test compound, both they compete for the binding to the hERG channel. So here you can observe this is the radioligand, these are the test compound, and these are the transfected cell. So this radioligand and this test compound, they both compete for binding to this potassium channel. Then after incubation, the membranes are filtered and they were washed with multiple times with a cold buffer. After that, if the test compound has a strong affinity to hERG channel, the radioligand response will be low, because here they have the affinity for the same channel. So if the test compound having the more affinity, the more compound will bind to this channel, and therefore the radioligand response you can observe is very low, and the radioligand response will be detected by scintillation counter, and the activity of the test compound is indicated by displacement of the labeled compound. So the concentration or the activity is based on the how much test compound is displaced by the radioligand compound has to be observed. So this is given that test compound has closed this channel and radio ligand are free in the solution, and they will not show any the response observed is very low. Then the next method, that is rubidium flux method. In this method, again the hERG gene, it is transfected with the Chinese hamster ovary cell. So Chinese hamster ovary cells are isolated and they are in fact transfected with hERG, and then they were pre-incubated in the media which consists of rubidium. So these Chinese hamster ovary cells are transfected with hERG gene, and then they were incubated in the media which containing the rubidium ion. Then after keeping this in contact or pre-incubated position, after some minute, the media is removed and the cells are washed with the buffer. So what happens? This rubidium ion, which are present extracellularly, they get washed out from the medium. Buffer containing the test compound and potassium ion is added to the rubidium containing medium and incubated. So after washing, the extracellular rubidium ion, they get washed out, and whatever the rubidium ion present into the medium, add the test compound and the potassium ion containing the buffer, and then again incubated for some time. So here, if the test compound blocks the channel, if the test compound block the channel, all the rubidium ion will stay inside the cell. And if the rubidium flows into the medium, it indicates the channel is not blocked by the test compound. If it is blocked, then rubidium will inside the cell. And if the rubidium are moving here, it indicates that the compound has not blocked the channel. Then the rubidium concentration is measured by either scintillation or atomic absorption spectroscopy you can use. Then next method is patch clamp technique. So this patch clamp technique, the instrument, first we see the instrumentation. The instrument comprises of the electrode and a glass capillary, microscope, amplifier, compound, recording software, micromanipulator, and a camera. An electrode, if you see in the figure, an electrode, it is inserted in the glass capillary, and this glass capillary is filled with the buffer. While observing the under microscope, here you can observe the Petri plate where the cell is kept, and you can observe all these things from the microscope, and here the current is change in the current is recorded. So here the tip of the micro electrode is placed on the membrane of the transpected cells that express the hERG cell. Then a negative pressure is applied by the capillary, and the capillary produces a patch which is tight on the cell membrane, and it forms a high resistance seal. Now further negative pressure is applied, a small portion of the membrane is pulled into the capillary tip and it is ruptured, forming a whole cell patch. So here in this figure you can observe how whole cell patch is looking. This is the capillary and this is the cell part which produces the whole cell patch. Then the potassium ion channel protein in the cell undergoes conformational changes, and the channel will be open in response to the applied positive voltage. The potassium ion moves out of the cell through the ion channel, creating an electric current. So potassium ion from this ion get outside, and as it comes outside, this electrode will measure the current or determine the electric current. And if the test compound blocks the hERG channel, the ion will not come out from the cell and no current will be formed, or there is a reduction in the current. So patch clamp can also monitor the ion channel event at millisecond level. So this is a very important methods for determining many movement or electrophysiology of the compound. Then Perkins fiber, this method, as you know, it is a ex vivo method which is used. So here in this case, as you know, Perkins fiber, it plays a very important role in the conduction and propagation of the electrical impulses located in the inner ventricular wall of the heart. And they are also susceptible to development early after depolarization and response to the fatal torsade pointa. Some studies suggested that many ventricular arrhythmia are actually initiated in the Perkins fiber conducting system. So here the isolated Perkins fiber can detect drug-induced effect on the action potential configuration and gives in-depth information about transient outward potassium ion current and the rapid slow forms of delayed rectified current. So here you can observe, here is the Perkins fiber, and we have to isolate this Perkins fiber for performance of this ex vivo test. So here in this case, first take the rabbit has to be euthanized and the heart removed and placed in potassium and glucose enriched extracellular solution which is oxygenated, and they are kept at the room temperature. Then after, the Perkins fibers are dissected along with the small pieces of ventricular muscle to prevent damage in the fibers. Then these fibers are divided into 2 to 3 pieces and pinned through the ventricular muscle to the silicon bottom of the experimental chamber, and The transmembrane voltage is recorded with glass microelectrode having a tip resistance of 10 to 2 milliampere. So by using this technique, one can also find out the hERG expression or potassium channel currents by Perkins fiber. Next technique is the ligand drop perfuse isolated heart preparation. So this is a experiment where the heart is removed from the animal animal body and then reverse perfusion via the aorta as shown in the figure. The ligand drop apparatus is itself is so quite complicated that instrument, and here you have to uh, isolate or monitor the isolated heart, and this allowed the heart to remain alive and beating for several hours. During this time, the test is drug is added to the perfusion solution. The perfusion solution will go from the heart. So in this perfusion solution, the test compounds is added and electrophysiological recording was carried out. And from that, it has been observed whether the QT interval is increases or changes takes place has to be observed. An electrophysiological study without the effect of the whole body can be recorded. Then next is the QT assay. It is the in vivo assay. Here QT interval or elevation of the QT progression of QT elevation is the hallmark for this assay. And here we can use the guinea pig, and guinea pig first anesthetized with a suitable anesthetic agent. Then the animal was placed on the heat pad with circulating water at a temperature of 37 to 39 degrees centigrade to maintain the normal temperature in the animal. Then mechanical ventilation is provided by the tracheotomy, and the monitor oxygen saturation by the pulse oximeter. The drugs is administered to monitor the blood pressure. The jugular vein and carotid artery were cannulated respectively. Regarding this particular part, I have already posted one video on the rat blood pressure, where you can observe how jugular vein is cannulated, how carotid artery is cannulated. Then ECG electrodes pins were placed on the limbs and chest of the animal, and ECG signals were recorded. After the drug administration, the animal was observed for 30 minutes to evaluate the effect of the drugs on the QT interval. Then another in vivo test is there, that is non-invasive telemetry. So the ultimate method for assessment and study of hERG block by drug candidate is ECG using animal. In this method, they produces the highly informative data such as the ECG tracers. Electrodes for various cardiac function are placed on the animals, and lengthening the QT interval and inducing Tarsidy pointer can be directly observed in the living animal in this manner. No need to do any type of the surgery. The method is definitive, but it is a labor intensive and expensive. So here you can observe this is the chamber where the animal can freely move, and they were connected with the certain devices which can directly receive on the monitor the change in the blood pressure or change in the ECG and change in the QT interval. So this is a non-invasive telemetry. Then some applications are there for this methods for the drug discovery process. So first is to review the screening hits and structures planned for synthesis using in silico tool to identify potential hERG liabilities based on the structure. Second, when material is available from synthesis or for compounds brought into the project by collaborators, screen them using a high throughput hERG assay. Then to perform a detailed study of the hERG blocking effect of selected advanced drug candidate using this process. Then perform ex vivo or in vivo arrhythmia telemetry study for pre-developmental drug candidation. So these are some applications of this hERG blocking methods in drug discovery. Thank you for listening this topic. So if you like, kindly share, subscribe, and like this video.